I'm going to be talking about a project that's been ongoing for about a year and a half. And as you can see, I'm going to be discussing EEG spectral power with relation to antisocial behavior in children and adolescents. And I'm going to parse down antisocial behavior into aggression and delinquency. So to begin, I'd like to ask us all to think about why antisocial behavior is important. And for those of us who are not psychologists, sometimes antisocial is misused in the general layman terms. It does not actually mean somebody who is a loner, somebody who doesn't want to hang out. It means behavior that is harmful to society, behavior that is anti-society. So we're talking about criminality, we're talking about drug use, we're talking about gang affiliation, bad behavior. And so this is interesting to all of us because we really have all been touched in some way or another by crime in the society in which we live, and by the effects of gang warfare, by drug use, other dangerous behaviors to the self and to society. In recent years, maybe the last decade or so, there's been a distinction made between aggressive antisocial behavior and non-aggressive antisocial behavior, to which I will refer as a delinquency from now on. And the rationale of this project is to try to better understand maybe some of the biological pathways to aggression and delinquency, as well as the commonality and the distinctions between them. So a little bit about aggression. The most dangerous forms of aggression that we might consider are probably things like fighting, weapon use, bullying, but in this study, this is a community sample, a lot more normative, a lot less criminality and psychopathology. The most commonly endorsed items are really arguing, teasing others, screaming, disobedience. So we're really not talking about extremely violent behaviors, but probably the low end of the spectrum on aggressive behavior. In terms of delinquency, we might think of things like vandalism, theft, petty crime, drug use, but in our study, we're mostly talking about lying, cheating, swearing, lack of remorse. In terms of aggression and delinquency together, it's important to note that aggression tends to decrease across development, and this is reasonable. We know that little kids might hit and push each other in kindergarten a lot more frequently than do adolescents, whereas delinquency increases across development. Also, this makes sense. We know that graffiti, that petty theft is probably a lot less common within really young children. Boys have a higher tendency of aggressive behavior, both delinquent and aggressive, across most studies. This is just a very common finding. And because I will be talking about genes and environment, let me just give you some baseline. Aggression is more heritable, 65% genetic, as opposed to 48% genetic. There is also what's known as shared environment that emerges in delinquency, but not aggression. This is an environment that is common to both twins, that does not differentiate their behavior, but that might make behavior develop in both twins in unison. And in terms of the correlation amongst these are maybe the things that distinguish aggression and delinquency, but they're highly correlated. Across studies, we found about 40 to 60% correlation between aggression and delinquency, and the same was true in our study. It was about an R of 0.6 in both genders and both ways that I'll be talking about. Now, the method I chose to use was EEG. This is not one of our kids, but it's very much a cap that would look a lot like the caps our kids wore. And I'm looking at two regions, parietal and frontal. Parietal examines P7, 3, Z, 4, and 8. And frontal, I have a harder time reaching, but FP1, FP2, AF3, AF4, and then F7, 3, Z, 4, and 8. So those are the regions I'll be talking about when I say frontal and parietal. Now, a little bit about why I chose to answer the question from the perspective I chose to answer it. Let's consider slow arousal theory. So what we're looking at here is three frequencies of brainwaves. The top one is beta, and we have here beta corresponds to wakefulness, concentration. They say excited, maybe, maybe not, but if you're paying attention to something, hopefully there's a lot of beta going on right now. And below that we have alpha waves. Beta is about 13 to 30 hertz in adults. It might be a little bit lower, 11 to 30 hertz in children. Alpha waves are below. That's about 8 to 13 in adults, 8 to 10.5 hertz in children. And that corresponds to a lower arousal state, probably a more relaxed state, but not as low arousal as theta, which is 4 to 8 hertz, which is really typical of drowsiness. So what is slow arousal theory? Actually, before I say that, I would like to just make the point that these are not mutually exclusive. It is not that one comes at the expense of others. All three of these frequencies, as well as other frequencies, are probably found at all times. It's a matter of how much proportion there is of each one. It's an important thing to keep in mind. So why slow arousal theory? Now, it emerged in the 50s that violent criminals, psychopaths, were more prone to slow arousal. What does this translate to? It might be kind of a bored brain state, someone who is seeking out risk, seeking out forms of dangerous arousal to really get the sort of arousal that most people just tend to have already. So this was found in the 1950s with exclusively male psychopaths and violent criminals. And in the 80s and 90s, this was examined further in children and adults, but again, almost all clinical and criminal cases, and almost all males. There were very, very few females before our study that really had this examined. The other thing that's interesting about some of these studies, for example, Dr. Rain, the co-PI on this project, looked at 24-year-old criminals in a sample that he has on an island called Mauritius, and went back and looked at their EEGs at age 15 and found that those kids had lower arousal, the ones who ended up being criminal. Other studies were a little bit parallel to this. It was kind of a retrospective look at the arousal early on. So there's kind of a longitudinal component there, where it seems like it might be predicted later on. It's not as clear whether there's a concurrent prediction. So these are all things that led us to ask several questions here, like, will this replicate? And I'll get back to that in a second. Principal components analysis, I don't really want to go on too long about this because it really is technical, but since I spent 6 months of my life learning how to do it with Saeed over there in the back from electrical engineering, I just wanted to give it a moment. It's a way to summarize the primary activation in the brain by looking at simultaneously several channels and saying, how do these really correspond to one another? How do we weight them so that we have the most accurate representation of the activation that's going on in a particular region? I can definitely go more into it if needed, but it is quite technical. So another thing that I should touch on before moving on is the basic model of behavioral genetics, and that is a comparison of monozygotic twins, what we might refer to as identical twins, and dizygotic twins, or fraternal twins. Now, the monozygotic twins share 100% of their genes, by definition. The dizygotic twins share on average 50%, like any other sibling. There's really no difference between them except for some of the environmental conditions, such as prenatal conditions, that might actually differentiate them quite a bit. So we use this baseline information with the correlations, the covariances and variances between observable traits to make estimates of the kind of influences on a given observable trait. So how do we parse this? In the models that I'll be looking at, we parse this into 3 types of influence. Now, A stands for additive genetics. So when we say genes, this is what we're talking about. We're talking about A. The C here is common environment that was shared between the 2 twins. That could be prenatal conditions. It could be watching the same movie when they were children. It could be particular parenting styles, even though that gets a little bit hazy because parents don't always treat children exactly identically, which brings us to E, which is environment that's not shared between the 2 twins. So I will be referring back again to A, additive genetics, C, common environment, and E, environment that's not common throughout the talk. So if there are any questions, I can clarify further. Just a moment about this study. This is the USC Risk Factors for Antisocial Behavior. It's an ongoing longitudinal study, currently in its 5th wave. Applying for a 6th wave, Dr. Baker has tracked the kids from age 9, 10 until currently they're about 20-ish. We have over 700 families of twins. It is a community sample, so again, not criminal, not clinical, and it's representative ethnically and in terms of socioeconomics of the LA area. Which this brings me to the purposes of my work. So the first aim was to answer the question, what is the role of genes and environment in the development of these 2 forms of antisocial behavior, delinquency and aggression? It's an important question because it kind of helps us understand how these 2 develop in use and as well as what differentiates them. It lays groundwork for looking at molecular genetic studies later on. It can answer some questions about whether it's appropriate to treat these 2 forms of behavior simultaneously in an environmental setting? It's an important question to try to understand how these things develop. I've talked a little bit about the sort of genetic and environmental influences on these things separately. In terms of together, there's about a 38% overlap found in the genetics of aggression and delinquency in a meta-analysis that took place about 2 years ago. And as well as a 60% overlap in common environment and a 10% overlap in non-shared or environment that's not common. The second aim, which I would say this is sort of the flagship aim of my project, so I'll go into it a little bit more, is to examine whether EEG spectral power can be considered a biomarker for aggression and delinquency. Now, as we've said in the past, it's been found in male violent populations. Is this going to generalize to a community sample? Is it going to generalize to females? Are we going to see the same sort of developmental portion? Is it going to be a predictive relationship rather than a concurrent relationship? In terms of hypotheses, we expected to see low arousal, so alpha and theta, predicting antisocial behavior. And because no one really in the past examined aggression versus delinquency, we didn't really have specific hypotheses about that, even though these were violent offenders in the past. And lastly, these relationships have emerged in the past. We expected to see something here, maybe not as robust, but something between low arousal and ASB, antisocial behavior. And so the question has never really been asked, what is the origin of this relationship? Is this primarily a genetic relationship? Is it primarily an environmental relationship? I was saying to my mother earlier, could it be that a child who was not shown enough baby Einstein is both under-aroused and more likely to be angry? It's possible that there's an environmental relationship rather than a genetic one.
It's a good way to put it for your mother.
There was no baby Einstein when she was born.
But the whole baby Einstein thing has been completely debunked.
Yes,
of course.
Yes.
So,
You're okay.
So, yeah, exactly. No parental guilt needed.
So the instrument I used for the first study here was the Child Behavioral Checklist, which is a gold standard of behavioral rating in children. It looks at a wide variety of symptoms, but we chose the two scales that were obviously most relevant here, aggression and delinquency. We looked at 2 age ranges. Wave 1 looked at kids who were 9 and 10 years old, the first wave when they came in, and wave 3, we chose mid-adolescents, 14 to 16. We also selected this wave because it was as large as the first wave, whereas the second wave had slightly fewer participants. Our method for this first examination is biometric modeling of twin data, which I went over very briefly a few slides ago. So let's just talk about mean levels for a moment, which we refer to as phenotypic rather than genotypic, so what's observable. We see that males are consistently showing higher levels in both aggression and delinquency in both waves than females, consistent with past findings. We do see that aggression decreases over the 2 waves, whereas delinquency increases. Again, exactly what we would expect, and just those are the means and the standard deviations. My mother asked about the ends, and so we have about 1,200 in both waves. Now, this is the findings. Notably, I'm going to explain this. Notably, the model that best fit the data had males and females be equal. So sometimes you get different estimates of genetics and environment on these observables that are different for males and females, not in our case. So this is a structural equation model, and the way to understand it is that the things that are measured are in squares, and the things that are estimated are in circles. I might be referring to latent factors. That just means it's estimated. So what we have here is that there's a latent factor of antisocial behavior that emerges both in wave 1 and in wave 3 that conjointly affects or jointly affects both delinquency and aggression in each of those waves. That means that a lot of the influences that we see jointly affect both of these sets of behaviors and are probably what contribute to that, not probably, they are what contribute to that correlation to what makes them similar. In addition, it's important to note that we have specific A and specific E that affect each of these subscales at each of these time points without affecting the others. So to go over it a little bit more, we have A, C, and E influences that all affect this latent factor, the estimated antisocial behavior factor in wave 1. And all three forms of influence also affect the wave 3 factor over there. The A affects both ASB1 and ASB3. And so we can understand that, and there's no A that emerges in wave 3, we can understand that all the genetics that affects the commonality between aggression and delinquency is there throughout this course of development. There's no new genetics that emerges in wave 3, whereas there is new common and non-common environment that emerge in wave 3. So there are new environmental conditions in adolescence that commonly affect both the tendency to be aggressive and to be delinquent. Another thing that I should explain here is the way that we look at the influence. These are path coefficients, so that they are squared in order to understand what proportion they're actually giving. So here, for example, we have 0.72 of the influences come from the ASB to the delinquency. If we square it, it's about 0.51-ish. And then A and E here are 0.50 and 0.48, about 0.25 each. So if we add this up, 0.50, 25, 25, it sums up to about 1. And this is the totality of the influences that we see on any observable trait. And the specific factors do not have any influence of C. So the things that differentiate aggression and delinquency from one another are not common environmental factors, but there is A and E influence that comes up. So just as a summary, we see common latent factors, so estimated factors of antisocial behavior, that are influenced by A, C, and E each. There's longitudinal stability, that is, the things that relate these two are A, C, and E types of influences. There's mu, C, and E that emerges in wave 3, but there's new environmental influence, both shared and non-shared, that emerges in wave 3, but all of the genetics on this common factor is actually shared between the two waves, so it is a major part of the stability between them. Time and subscale specific A and E also emerge, the things that differentiate aggression and delinquency across time. And again, males were, it was possible to equate males with females. That produced the best fit of the data. The second study was aimed at this AIM2, which, if you remember, was to see whether or not spectral power could be considered a biomarker for antisocial behavior. So we looked at, again, frontal and parietal. I showed you that on the earlier slide. Alpha, theta, and beta. Alpha, again, is a more under-aroused and relaxed state. Theta is an even lower arousal, kind of a drowsy state, whereas beta is, you consider, a more alert state. We did that principal components analysis to get the major activations in each of those frequencies, those two regions of the brain. I looked at aggression, delinquency, and CBCL. And the model here, or the method here, is multi-level mixed modeling. And what that means is that we take into account the fact that twins are correlated with one another. These are not independent observations. Being within the same family means that they're necessarily correlated. In fact, all of the behavior genetics is based on that. So this is a method to take a look at the effects of the family on the individual and try to get a relationship between two variables that takes into account the commonality between the two individuals. So just to show you what I appear to have a 0 there, I'm sorry. To show you what the graph is going to look like, we have frontal alpha, theta, beta, and then parietal alpha, theta, beta. Over here, wave 1, wave 3, females and males in each one, and aggression and delinquency in each one. I just wanted to just set it up before showing you the significant results. So I should have in the title here that what we're looking at is regression coefficients. And in the cases where things were significant, I also added the standard error in parentheses as well as the p-value. So these were all the significant relationships that emerged. There was no significance in wave 1 at all. In wave 3, we see that male aggression was associated with frontal alpha, frontal beta, and parietal beta, and it's a positive relationship. In wave 3, we also see that delinquency had a negative relationship. So there's a negative association there with frontal alpha and frontal parietal beta in girls and with parietal theta in boys. So this is, just to fill in, this is what the rest of it looks like. And those bolded and more elaborated upon numbers were the ones that were significant.
But just to be reminded, you expect from the literature positive relationships.
We expected positive relationships, yes, with at least with theta and alpha. And So we were surprised to find some negative relationships with delinquency. So just to summarize what we just saw, there were no significant relationships in wave 1. In wave 3, higher power in frontal alpha, frontal beta, and parietal beta were predictive of aggression in males, and lower frontal alpha and parietal beta, as well as in females, and parietal theta in males was predictive of delinquency. So I will elaborate a bit more upon what we think this might mean a little bit later, and move on to aim 3 just for the moment. We don't entirely know what the beta findings mean, and that is because it's really pretty novel. We have 2 studies in the past that looked at beta power and how it relates to alcoholism, and they found a positive relationship. So if we consider that a precedent, there's one precedent, but they weren't looking at aggression. So we chose to leave beta power out of the behavior genetics for now, and concentrate on frontal alpha power, because it's the measure that's probably been looked at most frequently in the literature, and there's more support for it than for some of the other measures. And we decided to look at the results of study 2 to decide what models would be examined in study 3. And if you remember, the frontal alpha relationships were positive with male aggression in wave 3 and negative with female delinquency in wave 3. So these are the 2 models that we're going to be looking at for aim 3. Male aggression, female delinquency, and the method here is a bivariate design. That means we're looking at just the genetic and environmental relationships between 2 observables. And these were the results for the males. We find that frontal alpha power is highly heritable. Here I squared it for you just to make a little more accessible what it means to have a correlation. So frontal alpha power was 78% heritable and 22% influenced by E. The C is no longer significant. And same with aggression in wave 14 to 15, there was no C that emerged here either, 67% heritable. Now, the entirety of the relationship between them, which was somewhat equal in magnitude to the phenotypic relationship, is accounted for by genes. The whole relationship that we observe is attributable only to the genetic influences, and it's a small correlation. It's the overlap is 0.22, but it was significant. That means that 0 was not part of the potential estimates there, so we know that there's probably some genetic correlation between these 2 observables. When you look at the females, again, the entirety of the relationship is explained by genetics. And this time it's a negative relationship, and that only corresponds to the fact that the phenotypic relationship was negative. And we find, again, that both delinquency and frontal alpha power were quite heritable. So what does this mean? We have small but significant genetic relationships. The phenotypic relationships that we observed in study 2 can be entirely explained by genes. They're small, but so is the genetic correlation that we find. And this leads us to ask some questions. Are we looking at a difference between aggression and delinquency? Are we looking at a difference between males and females? Why is it that these relationships seem to be going in opposite directions? Or is it entirely sporadic? It's always a possibility as well. And so considering what the potential problems may well have been, we came up with at least a few that we tried to flesh out a little bit with further analyses. For example, could it be a poor quality of EEG data? Could we be looking at differences between our community sample and past samples which were criminal or clinical? Is there a major difference between the populations to begin with, such that the relationship might disappear? Is there a problem with the CBCL instrument? Is this kind of a poor instrument for looking at antisocial behavior in children? So we set out to answer some of these questions. The first thing we did was try to examine the quality of the EEG data, and the way we chose to do that was looking at heritability. We figured if it's complete junk, there's not going to be the sort of correlation that we would see stronger in monozygotic twins than we do in dizygotic twins. But in fact, what emerged here was exactly what's been found in past studies, mostly in adults, never really in children of whose age with the eyes open. So in a sense, it is nice and novel. We're going to try to publish it. But we do find heritability. the scale here on the right demonstrates to us that blue would correspond to 0 heritability. We see no blue. The most that we see is about a 0.4, which corresponds to that white, and a 0.8 is the top red. We have males and females here. For beta, they were entirely equal. For theta and alpha, in the more posterior reasons, for whatever reason, females showed higher heritabilities. We're not entirely sure what this means, and nor is it, we're not entirely sure it's replicable, but it is an interesting finding. We might suggest that other people look into it. We're looking at here is 30 electives on their own. And so it doesn't seem to be that the problem is data quality with the EEG. Another thing that we thought about was that maybe these criminal and clinical samples are pre-selected from having experienced adverse environmental conditions, which we know is true. We know that people who end up more prone to crime, more prone to violence and severe psychopathology are more likely to have experienced difficult situations in their lives, adverse conditions, abuse, neglect. And we thought maybe we would try to see if there's a moderating effect of adverse conditions in our children. So we looked at several potential environmental moderators. We looked at victimization, socioeconomic status, neighborhood quality, and stressful life events. The table is far too big to actually show you. It's just overwhelming. But to summarize it, what we found was that there were 4 relationships that emerged significantly in terms of moderation relationships. This was also examined with mixed modeling. They were all in the males. And we found that adverse neighborhood plus higher frontal or higher frontal alpha or higher frontal beta led to higher aggression in the males. We also found that more stressful life events with lower frontal alpha or lower parietal beta contributed to delinquency. Now, this is actually notable because the lower frontal alpha was correlated with delinquency in females. And so we find that in males, this might also be the case if we choose males who've experienced no stress. So the number of relationships we actually looked at was pretty large. It's hard to say whether these are just spurious or not, but it's definitely worth further examination because it could be considered potential support for the distinction between delinquency and aggression in terms of arousal. We also thought, well, maybe the CBCL is not a great instrument. Maybe we should see if this generalizes to other instruments. We did find that the aggression replicated entirely using Adrian Rain's instrument, the Child Aggression Questionnaire. And what's interesting is this parses aggression into 2 forms of aggression, a more reactive and a more proactive aggression. Reactive aggression being the tendency to fight back when something is irritating or aggravating, and proactive aggression being a more instrumental form of aggression, being just likely to lash out at people for fun. And what we found, this might be a familiar pattern, that frontal alpha, frontal beta, parietal beta. It's what we saw using the CBCL as well. Emerged as a positive relationship in the males, and only with we do have that parietal beta with the reactive, but more so with the proactive than the reactive. And this is actually exactly what we would expect if the low arousal theory were the explaining factor here, because these would be the kids who are kind of bored and seeking out something to lash out on, rather than the kids who might be more high-strung and more likely to fight back reactively. This is just filling in the table here. Now, we did get 2 significant relationships in the females that weren't consistent with anything we found with the CBCL. We got frontal theta higher in conduct disorder, that's what CD stands for, and then CPS is the child psychopathy scale, so it's a personality measure of likelihood toward grandiosity, glibness, not caring for others, lack of remorse, lack of empathy. And, well, it's hard to interpret what these 2 would mean without a greater context. We saw only negative relationships in the females, but this is in fact closer to what we would expect. Then again, neither child psychopathy nor conduct disorder are purely aggressive or delinquent. One is a measure of personality, and the other one actually incorporates both forms of antisocial behavior. So it is a little bit harder to say whether this bears any distinction from the aggression-delinquency divide. I'm just filling in the table here. Okay, so we thought let's look further as to, at the genetic relationships to see whether we can find more support for that. So we decided to look at what's called a high-risk design, which is when we identify parents with a particular phenotype and look at their children to see if some sort of predicted biomarker would be observable there. So we looked at parental aggression and what we call non-aggression, we can call it delinquency as well, in both the mother and the father. And these were slightly different questionnaires, so it really came down to paternal aggression having only 4 items: frequent fighting, threats with weapons, assaults or attacks, and as you can imagine, very, very infrequently, I think it was a 0, endorsed murder or attempted murder. But paternal aggression was significantly associated with higher frontal alpha, frontal beta, and parietal. Oops, that's terrible. That's supposed to be beta right here. Sorry about that. Beta in daughters. So this is that same pattern that we saw before, both with the child aggression questionnaire and with the CDCL. So it seems like maybe there's something happening in these 3 regions, and it's not entirely clear what that is. It's also not entirely clear why these girls are not showing the, well, not these girls. That was something I thought about this afternoon. It's not entirely clear why girls are not showing this relationship. What I thought of was maybe these girls would. Maybe if I looked only at the girls whose fathers have been more aggressive, this would emerge. So that's potentially something else to look at. But it is that same pattern. But this is the only thing that we found. Maternal aggression and non-aggression was not predictive in wave 1 or wave 3.
Wow.
This is only predictive.
Good for us.
Good for you. Wave 3, and only for paternal aggression in the females. But it does lend some support maybe to the pattern that we saw in terms of genetics. So how would I conclude all of this? The aim 1 was to look at the structure of influences on aggression and delinquency. And we found support both for the distinction between them and for the commonality, the overlap between them. Over 50% of the influences on any observable trait came through a latent or estimated antisocial behavior common factor, and the other 50% were unique to that particular observed trait or behavior. The commonality accounted for about 25 to 48%. Oh, excuse me. The commonality was accounted for by about 25 to 48% genetic factors, and males and females showed equal influences. Aim 2, maybe the primary aim of this paper, the question that we really wanted to answer, was, is spectral power a good biomarker for antisocial behavior? And I have to conclude that it's not great. These were pretty small relationships. They were fairly sporadic. Certainly not the sort of robust low arousal and high antisocial behavior relationships that I was hoping to find having read all this literature. I don't think this is going to be a screening tool. But sort of the silver lining is, if this replicates, and that's a big if, it could be a really interesting biological distinction between aggression and delinquency. We found in the past that low arousal was related to violence, to pretty severe criminology. So, okay, can we say that aggression and low arousal are an association, and then delinquency and arousal? Is that possible? It is something that our data suggested, but certainly it would take a lot more replication for that to be something that we can kind of declare. And another thing that is worth considering is there a distinction between males and females? And males did show one instance and then another instance when moderators were considered where low arousal did correlate with antisocial behavior. But for the most part, what we found in the males was high arousal. Excuse me. So we did find one instance and then one with moderation where male high arousal did correlate with antisocial behavior, but what we mostly found was that it was low arousal, what we expected. On the other hand, anything we did find in the females was high arousal and antisocial behavior. So is it a fluke, or is it a fluke? Is it potentially something to do with delinquency versus aggression? Or are we kind of looking at a real sex difference there? Could it be that while antisocial behavior is more common for males, and thus a more under-aroused male might default to this behavior, it would take more to overcome the social barrier for a female to be antisocial? Could it be that it takes more planning, more concentration, more high arousal? It's possible. And so these are all things that should be further examined. What I should say, it just occurred to me right now, is that other measures in our lab of arousal also found much stronger support for low arousal and male antisocial behavior than female. So things like skin conductance, heart rate were much more correlated in males than it was in females. So this might actually be a legitimate sex difference that we're looking at. The aim 3 was looking at that genetic and environmental relationship between the frontal alpha power in this case and antisocial behavior. What we found, the patterns were consistent with the phenotypic findings. The magnitude was quite small, but the genetic relationships were significant. And the entirety of the relationships was explained by genes rather than environment. I would again say this is not a robust screening tool. This is not going to be something that we implement in schools to try to figure out which kids are most at risk. We did also have some additional support from that high-risk parental model with a paternal aggression. What are some developmental implications here? So just like in the past, we found that EEG was more predictive of later antisocial behavior than concurrent antisocial behavior. Could this be a marker of underlying vulnerability? Could it be that kids who show this less arousal are more likely to develop antisocial behavior rather than be currently typified by antisocial behavior? And another thing that we should consider is that certain theorists have suggested that there's adolescent-specific or adolescent-onset antisocial behavior, more so in delinquency than aggression, but there's definitely some evidence that these forms of behavior that are very much age-specific do exist, and so could it be that low arousal or high arousal are predictive of these particular forms? Further examination would be very helpful. So again, the rationale behind looking at environmental moderators was that our sample might be less extreme, and criminal or clinical samples might have been pre-selected for more difficult and adverse life circumstances. We found some hints, some trends toward support for this suggestion, but it could be that really what we would need to do to see if the moderation emerges was to look at more severe environmental conditions, so abuse or neglect. Could be that stressful life events is actually not enough to trigger the relationship between arousal and antisocial behavior, whereas more severe life circumstances would. So this is a potential future study. In terms of intervention implications, I think there are a couple of things that would be interesting to talk about. One is that we did have new environmental conditions emerge as significant influences in adolescence. I think this lends some support to the idea of socially focused or more integrated, community-integrated interventions for more delinquent, more aggressive adolescents. The other thing I would say is if this is replicated, the arousal is a very interesting target for intervention, either meditation or neurofeedback. And if we really do know that one of these forms of behavior is more correlated with one form of arousal, that's actually a very helpful interventional idea. What are some strengths and limitations of this study? Again, it's a community sample, so on one hand it's great to be able to answer the question, will this replicate in a community sample? But on the other, this meant that I didn't have that sort of robust phenotypic relationship with which to examine the genetics. It could be that if I had a huge population of twins who happened to be murderers, I would get a much more significant genetic relationship between EEG and antisocial behavior. It's a parent report. We wanted to be consistent, so we chose parent report because, again, it wasn't there in the first wave, but parents, inarguably don't know everything that's going on with their kids by age 14 to 16, so we could really be missing some stuff. It might be interesting to replicate some of this with that youth report. And largely, EEG itself is a bit of a problematic tool. There's a very low signal-to-noise ratio and a very high variance. It can be variable across time. Within hours, you can find a completely different profile. And so I've been somewhat disillusioned with EEG as a diagnostic tool. I think it might still have fantastic uses elsewhere, but perhaps not diagnostically. So in terms of future directions, I really want to hammer home this could be really interesting, this high arousal versus low arousal, delinquency versus aggression, if it replicates. If it replicates, it could be a really interesting biological explanation for this distinction between different forms of antisocial behavior. So let's talk about maybe taking it to criminal samples. What would happen if we looked at delinquent-only actual criminals, like white-collar criminals, or people who broke into homes but were never violent? And my mother asked, is there really a distinction between aggression and delinquency? Yes, there is. There could definitely be a very delinquent person who does not happen to be violent at all or aggressive at all. So that would be an interesting place to take this sort of research next. In terms of environmental moderators, as I said before, maybe if we looked at more severe adverse conditions such as abuse or neglect, some of these relationships might become more robust. Try to examine the origin of this sex difference more. Are we looking at an arousal problem or difference, excuse me, not problem, or is this all kind of fluky? And it would be interesting to continue this now that the twins are adults. We have EEG data from them collected at age 20 or so, and it would be really interesting to see if these sorts of relationships do endure long term. And lastly, look at the self-report. As I said, the parent report is probably limited. I believe I kept it under 45 minutes, and that's it.
You sure did.
Thank you.
Okay, thank you so much, Sharon.